Assalamualaikum. In the second part of the lecture, we will continue with the artificial vegetative propagation. So, artificial vegetative propagation usually occurs as a result of human activities. So, there are a couple of common methods of artificial vegetative propagation, which are by cuttings, layering, grafting, and also micropropagation, or also known as test tube cloning. So this is done by tissue culture method, or we call as plant culture. So methods of artificial vegetative propagation, the first one is cutting. So a vegetative part, this can be the stem, root, or leaf of a plant which is cut and planted in the soil along with some nutrients. So when supplied with the nutrients, the cut part will develop into a new plant. So for example of a stem cuttings, the plants are sugar canes, duranta, coleus, rose, and China rose. For root cuttings, the example are tamarind and lemon. For leaf cuttings, the example are peperomia, begonia, and African violet. So as we can see, for stem cutting, when the stem with some leaf is planted in the soil, the root will begin to grow into the soil. So this will grow into a mature plant. For root cutting, only some parts of the root is cut and planted in a soil. It will grow into a new plant. And for the leaf cutting, when a part of the leaf like this planted into a soil, so it will start to grow roots and eventually will grow into a mature plant. For micropropagation or test tube cloning, so in this method, we generate genetically engineered plant through tissue and cell culture technique. We call it as plant culture. So this is done in lab setting with sterilized environment. So in this method, a small piece of tissue is excised from a plant, sterilized, and then it is grown in a nutrient medium under aseptic condition. The tissues proliferate into an undifferentiated mass. So we call this as callus. So in this picture, it shows the callus or undifferentiated mass of the plant tissue. So this can be kept and multiplied for an unlimited period. And then the callus transferred into another medium with specific hormones. So when supplied with the hormones, the differentiation of plantlets is induced. So this is when the callus tissue transferred into next medium, which is supplemented with growth hormones. And then this plantlet is transplanted in a pot or soil and raised to maturity. So it will grow into a mature plant. So by using this method, there's a lot of number of plants, indefinite numbers, can be obtained from a small mass of parental tissue. Example of plants that use micropropagation is orchids. So this is plant culture lab. It is the environment needs to be kept sterile, so it is free from bacteria and fungal contamination. In this picture, it shows that the nutrient agar. where a small part of the plant excised is grow into a callus like this. There are several advantages and disadvantages of vegetative propagation. For advantages, the propagation of plant that produce little or no viable seeds. So it is important for the plant that produce no seeds because it can be propagated only by using stem, leaf or other parts such as root. So this method, vegetative propagation, can provide consistency in terms of quality without variation of the plants and help plants to spread. This enables survival of the following damage. Following the damage to parent tree. Even though the parent tree was burned or already fall, only if the leaf or stem survive, it can develop into a new plant by vegetative propagation. But there are also disadvantages of this method. Vegetative propagation causes the plant more susceptible to diseases because the entire plant may get affected as there is no genetic variation between the plants and no new varieties can be produced because all the plants are genetically identical to each other. So now we will learn about the sexual reproduction in plants. So most plants are capable of asexual reproduction, but they can also reproduce sexually. So the definition of sexual reproduction is the production of a new individuals or offspring by the fusion of egg and sperm which involve fertilization between male and female gametes. So gametes is a haploid, only half of the genetic copy. When egg and sperm fertilize, it will form zygote, which is diploid, 2N. So sexual reproduction results in offspring that are genetically different from the parents because it involves combination of genes from both gametes. Seed producing plants. So seed producing plants undergo sexual reproduction. Difference from the plant that produce limited that produce no seed or non-viable seed. Those plants use vegetative propagation, but seed producing plants, it will undergo sexual reproduction. And there are two main groups of seed producing plants. First one is angiosperms and the second one is gymnosperm. So as we can see in the figure, gymnosperm has the microscopic female gametophyte inside ovulate cone. So these plants has cones. This one is ovulate cone and another one is pollen cone that contains microscopic male gametophyte made by the sporophyte, which is the plant. And then in angiosperm, the microscopic female gametophyte is inside this part is called ovule. And then microscopic male gametophyte is inside this part is called anther. Okay, which form the sporophyte, 2N. So angiosperms are also called flowering plants. So they have seeds that are enclosed within an ovary. So this ovary usually a fruit. So fruits such as apple, right? When you cut the apple, you can see the seeds in the middle. So when you grow this, you plant the seed in the soil, it will grow into a new plant, right? So angiosperms rely on animals for pollination, such as birds. The vast majority of plants in the world are angiosperms. Besides reproduce sexually through the reproductive organ of the flower, angiosperm can also reproduce asexually. So in the figure, it shows different types of flower in angiosperms. Gymnosperms, however, have no flowers or fruit, but they have unenclosed or naked seeds on the surface of scales or leaves. So gymnosperm seeds are often found in cones. This is the picture of the cones. The seeds lies in between these scales. So this plant produce reproductive cones that contain the seeds that will eventually grow into new trees. So gymnosperm rely on wind for pollination. So evergreens such as pines, cedar, and spruces, as shown in this figure, are gymnosperm plants. So pollination enables gametes to come together within a flower. So in angiosperm, which is the flowering plant, the dominant sporophyte embryo produces spores that develop within flowers into male gametophyte, which is the pollen grains, as shown in this figure, and female gametophytes, which lies in the embryo sacs. So we will learn about the overview of angiosperm reproduction in part 3 of the lecture. Thank you.